Today's topic is hemoglobin. Hemoglobin was discovered by Hornfield in 1840. The role of hemoglobin in the blood was elucidated by the physiologist Claude Bernard. Hi, I am Manti. In this video, I will describe about its type, its significance, its clinical use, and its vital role in blood. Hemoglobin is an oxygen-binding protein found in erythrocytes that transport oxygen from the lungs to the tissue. Each hemoglobin molecule is a tetramer made up of 4 polypeptide globin chains. Each globin subunit contains a heme moiety formed of an organic protopropylene ring and a central iron ion in the ferrous state. The iron molecule in each heme moiety can bind and unbind oxygen, which allows for oxygen transportation in the body. The most common type of hemoglobin in the adult is HbA, which comprises 2 alpha globin and 2 beta globin subunits. Different globin genes encode each type of globin subunit. The 2 main components of globin synthesis are globin production and heme synthesis. About 98.5% of blood oxygen is bound to hemoglobin in RBCs. Each 100 ml of oxygenated blood contains the equivalent to 20 ml of gaseous oxygen. Hemoglobin is a tetramer allosteric conjugated protein. Each subunit contains heme prosthetic group. Heme is an iron-containing propyrin derivative. Hemoglobin is present in all vertebrates except Arctic fishes and in many invertebrates including annelids, many arthropods and some molluscs. A variety of hemoglobin molecules are produced in humans. All are tetramers. Hemoglobin has a quaternary structure, characteristics of many multi-subunit globular proteins. Most of the amino acids in hemoglobin form from alpha helices connected by short non-helical segments. Hydrogen bond stabilizes the helical sections inside this protein. It causes attraction within the molecule, folding each polypeptide chain into a specific shape. A variety of hemoglobin molecules are produced in humans. All are tetramers, consisting of a numerous combination of 7 distinct polypeptide chains, each encoded by a separate gene. For example, hemoglobin A1, which contains 2 alpha and 2 beta chains, which represents about 98% of all hemoglobin found in all adult RBCs. Alpha chain contains 141 amino acid residue and beta chain contains 146 amino acid residues. Thus, HbA1 is designated as alpha 2 and beta 2. The remaining 2% consists of HbA2, a minor adult hemoglobin. This molecule contains 2 alpha and 2 delta chains and is designated as alpha 2 delta 2. The delta chain is very similar to beta chain. During embryonic and fetal development, different sets of hemoglobin are found. One type of hemoglobin during early embryonic stage contains 2 zeta chain, which are very similar to alpha chain, and 2 epsilon chain, which are very similar to beta chain, whereas during fetal state, hemoglobin contains 2 alpha chain and 2 gamma chains, which are very similar to beta chain. The embryonic and fetal form have a higher affinity for oxygen. This is necessary in order to obtain oxygen from the mother's blood. Next, here you can see different Hb expression during different environmental stages of human. That is embryonic stage, that is less than 8 weeks, fetal, that is 3 to 9 month stage, and adult, that is from birth. One hemoglobin molecule binds 4 molecules of oxygen, 1 per heme. Oxygen and hemoglobin bind in an easily reversible reaction to form oxyhemoglobin. The reaction between hemoglobin and oxygen is an oxygenated, not an oxidation. Iron in ferrous state binds oxygen molecule reversibly, whereas in the ferric state it does not bind any oxygen. The most important factor that determines how much oxygen binds to hemoglobin is the partial pressure of oxygen. The higher the partial pressure of oxygen, the more oxygen combines with hemoglobin. So, when deoxyhemoglobin is completely converted to oxyhemoglobin, the hemoglobin is said to be fully saturated. When hemoglobin consists of a mixture of hemoglobin and oxyhemoglobin, it is partially saturated. The percent saturation of hemoglobin expresses the average saturation of hemoglobin with oxygen. For instance, if each hemoglobin molecule can bind the two oxygen molecule, then the hemoglobin is 50% saturated because each hemoglobin can bind a maximum of 4 oxygen molecule. The relation between the percent saturation of hemoglobin is elucidated in the oxygen hemoglobin dissociation curve. A plot of percent oxygen saturation of hemoglobin versus partial pressure of oxygen for hemoglobin will be S-shaped or sigmoidal. Next, the two main components of hemoglobin synthesis are globin production and heme synthesis. Globin chain production occurs in the cytosol of erythrocytes and occur by genetic transcription and translation. Many studies have shown that the presence of heme includes globin gene transcription. Genes for the alpha chain are on the chromosome 16 and genes for the beta chain are on chromosome 11. Heme synthesis occurs in both the cytosol and the mitochondria of erythrocytes. It begins with glycine and succinyl coenzyme A and ends with the production of the protopropyrin 9 ring. The building of an protopropyrin to a ferrous ion form the final heme molecule. There are few different forms of normal hemoglobin in human blood. The percent prevalence of each hemoglobin type depends on the stage of development. Next is about the synthesis of hemoglobin. There are numerous steps and processes which errors can rise. For example, heme synthesis involves multiple enzymes. There are multiple steps which involved in heme synthesis. For that we need 8 enzymes. Among them 4 are from mitochondria and 4 are from cytosol. The process starts in the mitochondria where ALA, that is aminolevulinic acid synthase, links glycerin and succinyl coenzyme A to form a ALA. In step 2 through 5 occurs in the cytosol. Next, aminolevulinic acid dehydratase takes 2 molecules of ALA and produces PBG, that is porprobilinogen. In the third step, porprobilinogen deaminase takes 4 molecules of PBG and produces hydroxymethylbilin. In the next step, europropyrinogen 3 synthase takes hydroxymethylbilin and produces europropyrinogen 3. In the fifth step, europropyrinogen decarboxylase takes europropyrinogen 3 and produces copropyrinogen 3. In the final third step of heme synthesis, which occur in the mitochondria, here the copropyrinogen 3 is then transformed to protopropyrinogen 9 by copropyrinogen oxidase. After that, the seventh step occurs when protopropyrinogen oxidase converts protopropyrinogen 9 to protopropyrin 9. And then the eighth and final step of heme synthesis is the addition of iron to protopropyrin 9 by ferrochelatase, which produces a heme molecule. So this is all about the synthesis of heme. So, hemoglobin synthesis is a complicated process. This takes place in the red blood cell. To understand, this is the summarized, which is in the diagram. Amino acids are assembled in the ribosomes and give rise to alpha 2 and beta 2 globin, and then transferrin gives iron, which will combine with the heme molecule. Heme molecule will have protopropyrin by the chemical combination of glycine and succinyl coenzyme A in the presence of vitamins B6 and form heme. Heme molecule consists of protopyrin 9 protein and iron. Protopropyrin plus iron is equal to heme. So this is the shortcut summary of hemoglobin synthesis. So next is about the role of hemoglobin for oxygen and carbon dioxide. Hemoglobin serves as the main vehicle for the transportation of oxygen and carbon dioxide. It carries oxygen from the lungs to the tissue and then carbon dioxide from the tissues to the lungs. Next, oxyhemoglobin is formed during physiological respiration when oxygen binds to the heme component of the protein hemoglobin in the RBC. This process occurs in the pulmonary capillaries adjacent to the alveoli of the lungs. The oxygen then travels through the bloodstream to the drop off at cells where it is utilized in glycolysis and in the production of ATP by the process of oxidative phosphorylation. It does not, however, help to counteract to decrease in blood pH. Ventilation or breathing may reverse this condition by removal of carbon dioxide, thus causing a shift up in pH. So, this hemoglobin exists in two forms, a taut form, that is T, and a relaxed form, which is denoted by R. Various factors such as low pH, high carbon dioxide and high 2,3 BPG at the level of the tissue favors the taut form, which has low oxygen affinity and releases oxygen in the tissue. Next is about deoxygenated hemoglobin. It is a form of hemoglobin without the bound oxygen. The absorption spectra of oxyhemoglobin and deoxyhemoglobin differs. The oxyhemoglobin has significantly lower absorption of the 660 nanometer wavelength than the deoxyhemoglobin, while at 940 nanometer its absorption is slightly higher. So, these differences is used for the measurement of the amount of oxygen in patient's blood by an instrument which is called pulse oximeter. These differences also counts for the presentation of cyanosis, the blue to purplish color that tissue develop during hypoxia. Next is about the Hb role in oxygen. Hb consists of protein portion as globin and iron part as heme. Iron combines with oxygen and gives it a red color, whereas oxygen combining capacity is directly related to Hb concentration and not as the number of RBCs. Oxygen does not dissolve easily in water, so only about 1.5% of inhaled oxygen is dissolved in blood plasma, which is mostly water. About 98.5% of blood oxygen is bound to hemoglobin in RBCs. Each 100 ml of oxygenated blood contains the equivalent of 20 ml of gaseous oxygen. Next, one hemoglobin molecule binds 4 molecules of oxygen, that is 1 per heme. Oxygen and hemoglobin bind is an easily reversible reaction to form oxyhemoglobin. This reaction between hemoglobin and oxygen is an oxygenated but not an oxidation. Next is about the sign and symptoms due to Hb levels. A low hemoglobin level is often referred to as anemia. Causes of anemia may include anything that interfere either with the level or the function of hemoglobin or RBCs. Causes of low hemoglobin mainly include blood loss, loss of production or hemolysis. Blood loss may occur due to surgery, heavy menstrual periods, bleeding in the gastrointestinal tract or any other forms of bleeding. Premenopausal females are more likely to have a low hemoglobin level than men due to menstruation. And then due to lack of production also HB level may decrease. It may be due to the diminished production of the cells in the bone marrow or may occur due to bone marrow failure. It may seen at the case of aplastic anemia or infiltration of the bone marrow with cancer such as leukemia, lymphoma or solid tumor such as metastatic breast cancer. Low level of hemoglobin may also seen due to nutritional deficiency. It may be due to iron deficiency, vitamin B12 deficiency or folate deficiency. It can also cause anemia. It can be seen in case of kidney diseases also, which may be due to the release of erythropoietin. It is a hormone that promotes RBC production. Next is about the conditions with high HB. It may be seen in case of lungs diseases such as COPD or pulmonary fibrosis, or it may be due to congenital heart disease or due to right heart failure. In these cases, the increased hemoglobin is the result of the body's compensatory mechanism for low oxygen. A hemoglobin test may show an artificially elevated hemoglobin level due to hydration. In this situation, hemoglobin is more concentrated when the fluid in the body is low. Hemoglobin may also be elevated in people which living in high altitude due to the oxygen in the atmosphere. Next, condition in which hemoglobin has an abnormal structure which includes sickle cell anemia or thalassemia. Sickle cell anemia is an inherited condition in which abnormal hemoglobin result in RBCs shaped like sickles. They can get stuck in blood vessels which results in pain, blood clot and increased stroke risk. Next, thalassemia is an hereditary disease are characterized by different types of abnormal hemoglobin. Both alpha thalassemia and beta thalassemia have many different types and symptoms may vary from none to severe. People with thalassemia have often lifelong anemia and may require frequent blood transfusion. Thalassemia intermedia is also termed a non-transfusion dependent thalassemia and may not be discovered until adulthood. So this is all about the sign and symptoms due to HB level. So this is all about today. Today I have discussed about the structure, function, synthesis and the types of hemoglobin. Thank you for watching this video.